G'day everyone, thanks for joining me. Remember that before we get into some of the fascinating historical content surrounding the discovery of major world-shaping medicines, we're spending a bit of time laying a basic scientific foundation to appreciate the science of pharmacology, the effects of drugs on the human body. In this particular episode, we're going to build on our recent studies by looking at how the human body uses enzymes to change the chemical structures of drug molecules to make them less greasy and more easily removed by the kidneys. That might seem a bit of an obscure topic to consider early in this lecture series, but I hope to convince you that this is a really important phenomena that has a lot to do with how effectively drugs work within humans, but also some of the problems that accompany their use are often traced to their effects on drug metabolism. Okay, so let's just refresh our memories of where we've been in the last couple of episodes. Remember how we looked at the all-important process of absorption of drugs, which begins after pills are ingested. They undergo disintegration and dissolution to release individual molecules that then transition from the fluid within the upper intestines into the bloodstream, and then they get distributed around the body. Now we come to the process of metabolism, which tends to counteract the penetrance of drugs into tissues. So to see the significance of this phenomenon, remember that we've been looking at the all-important drug concentration in blood versus time curve. It's a really valuable piece of information whenever we think about the pharmacology of any drug. After we ingest a couple of pills, for example, they release their pharmacological chemical payload, and we get this process of absorption whereby the concentrations of the drug increase in the blood, and then they reach some sort of peak due to at which point that rise in blood concentrations tends to be counteracted by drugs penetrating into various tissues and being accumulated in tissues of the body. So that rise in blood concentrations comes to a peak, but then it begins falling. And this is under the influence of metabolism, but also another phenomenon we'll look at in a subsequent lecture on excretion. But metabolism is particularly important, and it has a lot to do with how quickly the blood concentrations fall with time after passing the peak, after ingesting a new dose of a drug. Okay, so how readily and how much affinity a drug has for a metabolic pathway has a lot to do with how quickly blood concentrations fall, and that determines how often you need to give fresh doses or new doses of the drug, whether you give it every 8 hours, every 12 hours, every 24 hours, every 48 hours, has a lot to do with how efficiently the drug is metabolized by enzymes in the liver. Okay, so we can define this all-important process of metabolism then as a process whereby absorbed greasy drugs sustain changes to their chemical structure during processing by drug-metabolising enzymes, or DMEs, in the liver. You may never have heard about these amazing little proteins, but I think they're some of the most amazing structures on the planet. They're certainly some of the most economically important if you look at how much money the pharmaceutical industry invests in studying the ability of these enzymes to metabolize new drugs. It's just a recognition of how important they are in determining the overall effectiveness of drugs within the body. Okay, so DMEs, these drug-metabolizing enzymes, are typically sort of mid-sized proteins that are located usually in the cell membranes in the interior of liver cells. Okay, this is a pretty important location because it means that any greasy molecule, any greasy drug that gets into the liver is going to typically accumulate within these greasy membranes within the interior of the cell. And this means that they are right where they need to be, right next to the drug metabolizing enzymes that protects the body by metabolizing and detoxifying these substances. Okay, so the drug metabolizing enzyme contains an inner chamber, catalytic chamber or active site, which carries out this complex chemistry that converts a drug into a more water-soluble metabolite, often by sticking an oxygen and a hydrogen, the so-called hydroxyl group, onto that drug to make it a bit more water-soluble. Okay, so this is a really important capability that the body has, this ability to protect itself against foreign substances that are greasy and otherwise have a tendency to accumulate in the body. Because if you think about it, if we just keep taking greasy drugs, and remember that medicinal chemists and pharmaceutical chemists design drugs to be greasy so that they can get out of the gut and get into the tissues of the body where they need to produce an effect on some sort of disease process. But the problem with that, if we keep taking repeated doses of these greasy molecules, they may well accumulate in the tissues of our body to the point that they start causing toxicity and harm to that tissue. Okay, so this is a bit of a problem. There needs to be a way for the body to protect itself against chemicals of this kind. The solution then is to convert these greasy molecules into more water-soluble metabolites. And this is where these all-important drug-metabolising enzymes, particularly within the liver, are so important. They convert the drugs into these more polar or water-soluble metabolites, which can then be removed by the kidneys and eliminated in the urine. Okay, so this is a pretty important capability that the body has, because it means that with repeated dosing of drugs, they don't just accumulate in tissues and cause harm, but rather the body quickly removes them, and this allows levels to stay within safe thresholds within that therapeutic window within the body. Okay, so this is a positive capability that the human body has that's so important to human biology. Now, if we had time, we could look at a lot of the science underlying the field of drug metabolism. It really is a huge field of study. And in fact, we've known about these phenomena really right back since about the middle of the 19th century, when researchers in Germany first discovered them during their very simple original experiments in this field. We've come a long way in the last 150 or more years, and we now know a lot about hundreds of drug metabolizing enzymes that are distributed throughout nature, many of which are present in the human genome, and they're there to protect us against foreign substances. Most of them, as you might expect, are strongly present in the gut wall, in the upper intestines, but especially the liver. And these are the two organs that are so important because they're the first port of call for any chemicals in the foodstuffs that we consume. They immediately make their way to the gut wall and the liver, and it's important that we have protective capabilities in those tissues to render those compounds less toxic by metabolizing them. But also some of the other tissues of the body also have some capability to metabolize foreign substances, including drugs, and that includes the brain, the kidneys in particular, the lungs, a few other tissues as well, but we won't go into that. Really, from a pharmacology perspective, most of the drug metabolism that happens in humans happens in the liver and the gut wall. Now, also, while we're focusing on pharmaceuticals in this lecture, it's not just these processes don't just protect us against pharmaceuticals. So while we call it drug metabolism, bear in mind that these enzymes also protect us against the tens of thousands of chemicals that we can potentially encounter in the modern world, both pollutants that enter the body from the environment, or industrial chemicals that we might be exposed to in the workplace if we work in particular occupational settings that handles chemicals, or if we inhale air in an urban environment. We're often inhaling products of the combustion of tobacco or of fossil fuels, all of which need detoxifying by the body if we're to be protected against them. Even substances in our food that enter via agricultural practices into the food chain, or substances we might use in the home, in the bathroom. Cleaning agents contain chemicals, or in the garden shed, herbicides, pesticides, solvents and paints all contain chemicals that potentially can enter the human body, and we need a way to protect ourselves against them. And this is what these remarkable drug metabolizing enzymes do. So I like to think of them as a very important complement to the immune system. In the last couple of years, haven't we heard a lot about the immune system? We've heard about T cells and B cells and antibodies and all sorts of components of the immune system. It's very important in protecting the body against big threats, against bacteria, against viruses, against fungi, all of which are quite big structures. But what I'm talking about today doesn't get as much glory, but it deserves it, because these capabilities that we study in pharmacology protect us against the potentially tens of thousands of substances that are small and can fly under the radar of the immune system, and we need a way for the body to protect us against those sorts of chemical substances. Okay, now you may be the sort of person that doesn't particularly enjoy chemistry, and so my commiserations, but I do need to cover a little bit of chemistry, and I'll try to keep it as simple and understandable as possible, just to show what is achieved by these drug metabolizing enzymes and how they protect us against foreign substances. And generally what they do is they convert greasy molecules into more water-soluble ones that can be removed by the kidneys. But let's just look at how they achieve that. Now, we can consider a very common feature of many drug molecules is that they contain what we call an aromatic ring, and particularly what we call a six-membered aromatic ring. And this is a benzene ring containing 6 carbon atoms, the element of life. And these carbon atoms in a benzene ring share a feature known as aromaticity, and we indicate that with a couple of 3 double bonds and 3 single bonds, but in effect these electrons are shared all around that structure. So these are very flat, stable, what we call planar molecules, and this confers a lot of solubility, on a lipid solubility on a drug. So this is why medicinal chemists include these sort of rings on drugs, because it tends to improve their penetrating ability to get into the body via absorption, but it also often enhances their binding to drug receptors and producing their pharmacological effects. But we're getting ahead of ourselves because we'll look at that in a future episode. Now, what happens though is that because so many drugs contain one of these aromatic rings, a very common occurrence during drug metabolism is what we call phase 1 metabolism or oxidative metabolism, because it oxidizes a drug by sticking an oxygen and a hydrogen atom onto that aromatic ring. Okay, so this new hydroxyl group, as we called it, an oxygen and a hydrogen, is very frequently associated with processing of pharmaceuticals by phase 1 enzymes in the liver. They generate this hydroxyl group. Now, that tends to confer a little bit more water solubility on the molecule. So this metabolite has a little bit better water solubility than that parent drug. So it's a little bit less greasy. But then in the second round of metabolism, and this is what phase 1 metabolism sets the drug up for really, it sets it up for what we call a conjugative reaction. And in a conjugative reaction, some existing molecule in the cell, and very often it's a sugar, and sugars are very polar, highly water soluble. They've got lots of oxygen atoms stuck on the sugar ring. This example shown here is meant to be glucuronic acid, and it's a very common addition to a drug during metabolism within the liver. So conjugative enzymes very commonly attach glucuronic acid, a 6-membered sugar ring, not too different from glucose in terms of its chemical structure. They commonly conjugate those or stick them onto a drug molecule. And the resulting product, this so-called glucuronide drug metabolite, is often very, very water soluble indeed. And so these metabolites are very quickly removed from the body by the kidneys. Okay, so this is a really important capability. And just to see how that gain in water solubility occurs, let's just spend a little bit of time. Let's get rid of these labels here, to get rid of the labels on the screen, and instead replace them with water molecules. Okay, so these little blue structures here, H2O, 2 hydrogens attached to an oxygen molecule. And because oxygen atom, because oxygen atoms have what we call electronegative properties, that is, they tend to hog electrons, and they pull electrons away from any atom that's attached to an oxygen. And in the case of water, this means that the oxygen pulls electrons away. Electrons are negatively charged, so it makes the oxygen a bit more negative and the hydrogen a bit more positive, a bit less negatively charged, so it becomes positive. Okay, so now this is a really important feature. In fact, many people say, the so-called fine-tuning argument, that if the hydrogen bonding properties of water molecules were any different at all than they are, life on Earth wouldn't be possible, because they're just the right strength for water to exist in 3 states on Earth, as ice, liquid water, or vapors or gas. If the hydrogen bonding interactions between water molecules was any stronger, then all the water on planet Earth would be frozen. And if it was any weaker, all the oceans would evaporate and exist as vapor. Okay, so it's just right. Now, these sort of considerations are also important when it comes to pharmaceutical science, because the body is predominantly full of water. And so we need drugs to dissolve in water if they're going to get metabolized, or rather transported around the body. But then also, from the point of view of protecting the body during drug metabolism, the body needs the capability to convert those greasy, less water-soluble molecules into more water-soluble molecules. And this is what these phase 1 and phase 2 reactions are doing. Because if you look at the parent drug here, there's very little opportunity for hydrogen bonding. Although those carbons on the benzene ring have hydrogens attached to them, they're not really electrically active. They don't interact much with water molecules at all. So this is why the parent drug is often very greasy and has low water solubility, because it just doesn't interact chemically with via hydrogen bonding with water molecules in solution. So this will be hard to dissolve. But when we look at that first phase 1 metabolite, remember there's this oxygen and a hydrogen group stuck on the drug. This can now start forming these transient hydrogen bonds, or these transient interactions with all these molecules of water swimming around the drug. Now, this is a very simplistic diagram. In reality, the molecule is completely swamped by water all around in 3 dimensions of the structure, and the water is continually jiggling around and moving. But I've just shown this very static, simple representation. But you can see the oxygen and the hydrogen attached to the drug are both undergoing hydrogen bonding with adjacent water molecules. So this means that this metabolite is just a bit more water soluble than the parent drug. But then when we look at the sugar, all these sugar molecules are bristling with oxygens and hydrogens attached to them. And this allows a lot of hydrogen bonding between adjacent water molecules and the sugar metabolite of the drug. Okay, so this confers a really high degree of water solubility on the molecule because of all these positive favorable interactions between water molecules. It means that the drug is sort of accommodated in solution in water to a degree that the parent drug just simply can't be. Now, if we want to see that in a simplistic form, remember how I have been using this idea of an octanol water biphasic system to explain how grease-loving versus water-loving a molecule is, how lipophilic, grease-loving versus hydrophilic, water-loving it is. Remember how we said that we take a flask, this is a traditional method, and we add equal volumes of water and a slightly greasy alcohol, octanol, and they don't mix, so they float. Octanol floats on the water. We add a greasy drug, close the vessel, give it a good shake, make sure it's all dissolved, allow the layers to separate out, and we find the greasy parent drug tends to go mainly into the upper organic layer. So this is representing the tendency of that molecule to penetrate greasy cellular membrane environments. But then after that drug undergoes a single step of metabolism, oxidative metabolism, a phase 1 reaction in the liver, remember we form this typically a hydroxylated metabolite with an oxygen and a hydrogen attached. Now this has become a little bit more water soluble. Remember, it has limited opportunities for interactions and formation of hydrogen bonds with adjacent water molecules in the vicinity of the molecule, but it's not great, but it does give a bit better water solubility than the parent compound, and we see that in this sort of octanol-watanol experiment. Most of the metabolite is still up in the greasy layer, but there's some also present in the water layer. But then after the drug has the sugar molecule attached in a phase 2 conjugative reaction, also carried out in the liver, this is where we see a huge gain in water solubility because of all that hydrogen bonding between the sugar molecule and the water molecules. This is a highly water-soluble substance. So now this is usually entirely hydrophilic and there's very little drug left in the octanol layer. Okay, so this is the goal of metabolism then, because once this metabolite acquires this high degree of water solubility, it's then ready to be eliminated in the urine by the kidneys. Okay, so this is how the liver and the kidneys cooperate to protect the body against foreign substances, particularly greasy ones. The liver converts those molecules chemically through the action of drug-metabolising enzymes into less greasy, more water-soluble metabolites, which are then removed by the kidneys into the urine. Okay, so that's the common fate of most drugs in the body. We're going to look at this in more detail in my next lecture, but that's the most common scenario for getting rid of drugs in the body: metabolism in the liver, removal of metabolites by the kidney. Now, as I mentioned, there's more than just one drug metabolizing enzyme in the body, and there are many of them, dozens and dozens of them in the human genome. And because of these multiple pathways of metabolism, we actually can sometimes get quite complex patterns of metabolism, a large number of metabolites being formed from a single drug. And in this scenario, we've got just only 3 enzymes having a go at a particular parent drug. Here we've got a greasy molecule, and it makes its way into the liver, and DME1, drug-metabolising enzyme 1, for example, converts it into a metabolite. DME2 converts into metabolite 2, DME3 generates metabolite 3. But then what we often see is some of those substrates then go back and get further metabolized by another drug metabolizing enzyme. So in this case, metabolite 1 then becomes a substrate becomes converted to metabolite 4 by drug metabolizing enzyme 2, whereas metabolite 2 here is now getting processed by enzyme 3 to form metabolite 5. In actual fact, it can get a lot more complicated than that, with a lot more than 3 enzymes participating. And I think it's quite common for a lot of pharmaceuticals to form a dozen or 20 metabolites. Is not unheard of. In fact, I think the record is probably held by chlorpromazine, a drug that's used in treatment of serious cases of mental illness. I think over 150 metabolites have been detected in patients treated with chlorpromazine, just because there's lots of chemistry going on during processing of that lipophilic molecule in the body. But that's a bit of an exception, but nonetheless, it just shows that sometimes we can get very complex metabolism and a wide range of metabolites being formed from a single drug as a result of these broad metabolic capacities of the human liver. Now, I just want to quickly wrap up this video by going through 4 areas of medical significance regarding metabolism, because you might be sitting there thinking, what does this really matter? So what if the chemical structure of a drug changes as it goes through the liver? Why does that matter? And why do we have to think about that in a context of medicine? I just want to give you 4 quick reasons. And the first one is that usually being processed by the liver and converted to metabolites just simply decreases the amount of time drugs spend in the body. So the more efficiently and rapidly a drug's metabolized, the more often you have to give repeat doses. Now, I could make this point in lots of ways, but I thought I'd just refer to a phenomena that's really quite significant and perhaps should be getting more attention than it does at present, but the phenomenon known as pharmacogenetics. And it turns out that every one of us carries variant forms of some of these drug metabolizing enzymes. And we sometimes see the implications only when we're treated with a particular drug that gets metabolized by that drug metabolizing enzyme. It's possible for us to go through our lives without having any knowledge whatsoever that we may lack a particular enzyme that needed to metabolize a particular drug. And it's only when we're treated with that drug that we might see unwanted effects. So I've got a simple scenario here where a couple of ladies were showing the impact of genetic variants with regards to drug metabolizing enzymes and how it influences their response to pharmaceuticals. So here we've got a lady called Kitty. She's got 2 functional copies of the gene. Okay, so most of the time we have 2 copies of every gene, obviously. Get 1 copy from mum, 1 from dad usually. And in this case, this lady has 2 functional copies of a drug metabolizing enzyme gene. So she's able to fully metabolize a drug in the expected manner. But her friend Jenny, and perhaps she's of a different ethnicity, and this is what we sometimes see, certain genetic traits affecting drug metabolizing enzymes tend to be present in higher frequencies in different ethnic groups. But this particular lady has 2 variant genes of the particular drug metabolizing enzyme. So she actually can't make that enzyme. So she can't metabolize certain drugs via this pathway. So let's see what happens. We administer the same drug to both individuals, and Kitty takes the drug and it gets converted by the drug metabolizing enzyme to its hydroxylated metabolite. And if we look at the plasma concentration time curve, so she's getting 2 pills here, we see this rise in blood concentrations, and as expected, it reaches a peak, falling within the therapeutic window, and then the blood concentrations fall as a function of time as a result of metabolism and removal of the drug from the blood as it goes through the liver. The drug gets chomped up basically by this drug metabolizing enzyme. Okay, so let's say when we put a time scale on this, that this drug has a half-life of 3 hours, and that just means that blood concentrations of this drug in kitty decrease or fall by 50% every 3 hours. Okay. Now let's see what happens when we administer the same dose of the same drug to her friend Jenny, who lacks these metabolic capabilities. So in this case, I've shown grey here because Jenny just doesn't make that enzyme. She doesn't have this capability to convert the drug into its metabolite. So when we look at her plasma concentration time curve, she's received the same dose of drug as her friend Kitty, but you can see the blood concentrations rise very significantly from the same dose and exceed the therapeutic window and are now up here in this range causing toxicity. Okay, so this is why you hear a lot of talk these days about personalised medicine and the idea of gene-guided drug dosing. The idea is that hopefully not too far away we'll all be able to get our genome sequenced, and we'll be able to work out which particular variant drug metabolizing enzyme genes do we carry or do we inherit from our parents, and therefore it might help in selecting drugs that are safe for individuals. And this is just a recognition that we can't just give the same dose of the same drugs to every person in the population and expect the same outcome. Sometimes there are genetic factors involved in an individual's response to drugs. And in this case, the lack of a drug metabolizing enzyme shows how important these enzymes are in normally controlling how long drugs spend in the body. When we can't metabolize them, as in the case of Jenny here, they stick around for a lot longer and cause problems. Okay, so we'll go over this more quickly, but another consequence is that usually drug metabolism means that drugs don't produce the same pharmacological effects once they're converted to a metabolite. So I'm getting ahead of myself, but we're going to talk about this in coming up lectures on pharmacodynamics, how drugs work. But essentially, most drugs work by binding to some sort of protein in a tissue that controls the function of cells in a particular tissue. So let's say this is a heart drug, and it's binding to a receptor in heart tissue. And so the drug is carefully designed to fit certain binding sites where the drug docks or binds to a particular receptor and produces some change in that receptor that changes the function of the cell and the tissue. And let's say in this case, we see a decrease in blood pressure as a result of the drug binding to this particular receptor. Now, what happens when the drug gets metabolized is we see now there's this sugar group stuck on it. And so now this drug simply can't fit into that binding site anymore. It just doesn't bind anymore. So in this case, the metabolite doesn't contribute at all to the pharmacological response. So usually when a drug gets metabolized in the body, it switches off its biological activity, and that's one of the reasons why we need to replenish the dose with a fresh dose. Okay. Now, but there are exceptions to this scenario, and sometimes when pharmaceutical companies are working on a drug that seems quite promising, they make the disappointing finding that the drug has poor oral availability. Remember, we talked about this in an earlier lecture, how that sometimes drugs are just too charged or too water-soluble to be well absorbed from the gastrointestinal tract and make their way into the bloodstream. Now, sometimes what the chemist can do is convert this molecule into what we call a prodrug. And a prodrug is one where they have made a chemical change to the molecule and they've stuck some sort of greasy group of atoms onto the drug. And then they rely on the fact that after the drug now gets absorbed into the body, because it's greasier and able to get into the body, it then gets converted by an enzyme, a drug metabolizing enzyme, which chops off that greasy group and liberates the parent drug as well as the greasy group. Okay, so in this case, a drug metabolizing enzyme is converting an inactive prodrug into an active pharmacological agent within the liver. You might think, oh, who cares about this? Why am I talking about this? But this is actually becoming quite important because about 1 in 6 drugs or so nowadays that get approved in the modern world are based on a prodrug strategy. So this is becoming more important. Okay, and one reason why, to wrap up now, the last issue we want to talk in terms of metabolism, why we need to know a little bit about prodrugging, is because sometimes other drugs can mess up the metabolism of prodrugs, and so they rob patients of the benefits of those drugs. And let's look at this scenario here where, let's say we've got a patient taking a drug. Let's say they suffer from some chronic condition, heart disease, and they need a blood pressure lowering medication for years and perhaps decades of their lives to keep their blood pressure down. Okay, so in this case, this drug A, this blood pressure lowering drug, is normally metabolized by a drug metabolizing enzyme to generate a metabolite. But what happens though, this patient, perhaps they go off to, Mexico or Bali or wherever and have a holiday. They come down with some sort of food poisoning episode, they've got an infection. The doctor prescribes an antibiotic, drug B, and doesn't take enough time to check the medical history of this person. And it turns out that drug B, the antibiotic, messes up the metabolism of drug A by blocking the active site and preventing drug A from being converted into a metabolite. We call this drug B an inhibitor of the DME. And this is a really common problem in modern medicine because of the phenomenon of polypharmacy, the use of multiple medicines simultaneously. You have to be very careful that we're not administering a drug that slows down the metabolism of existing drugs in the patient, because this can result in dangerous increases in blood concentration, so that safe doses of drug A may actually start causing problems just because we co-administered another drug that blocks the metabolism of drug A. Very common problem. This is one reason why the world needs pharmacists, because one of the jobs of pharmacists is to pay careful attention to the existing medical drug history of patients and making sure they know exactly what pharmaceuticals are being taken and whether or not drugs with inhibiting properties are being combined with inappropriate ones. But it's not just of course, drugs that can have this effect. We know more now about fruits and fruit juices, their ability to mess up drug metabolism. Of course, one of the most famous examples is grapefruit. I don't know why anyone would want to eat grapefruit at the best of times, but it can inhibit metabolism of many, many drugs. So you've got to be careful if you're taking medication that is affected in this way, not to consume grapefruit. Okay then, I've been through a lot of ground in this lecture. Hopefully it's given you a bit of an appreciation for how important metabolism is in shaping the overall responses of people to drugs. But let's quickly recap what we've looked at. Firstly, we've seen that metabolism protects the body by converting greasy small molecules into water-soluble molecules. And as far as I'm concerned, it's pretty well just as important as the immune system in providing low-level protection against incoming substances that have potential to cause bodily harm. Secondly, we've seen that the remarkable catalysts that carry out this chemistry, drug-metabolising enzymes, are usually able to metabolize many different drugs and can form many metabolites. Okay, so this is really quite remarkable that just a small number of enzymes are responsible for protecting us against literally hundreds, if not thousands, of drugs. Thirdly, we've seen that metabolism usually terminates the pharmacological effects of drugs, but sometimes it can actually make them more active, and we particularly use that capability when we're dealing with prodrugs. And then finally, care must be taken when using drugs in people that have inherited gene variants. So we call that pharmacogenetics and personalised medicine. We need to be careful that we don't give excessive doses of drugs to people who can't metabolise those drugs via a particular DME because they've got an inherited deficiency in that pathway. But also, more commonly perhaps, there's a problem raised when we give drugs that block certain drug metabolizing enzymes along with other drugs that depend on those enzymes for being removed from the body. We have to be very attentive whenever we prescribe multiple medications to the same patient. Okay, I think that's pretty well all I have time for in this particular session. Thanks again for joining me. Hopefully you'll come back to see my next episode, which will wrap up our little consideration of pharmacokinetics, and we're going to look at E is for drug excretion, which is an alternative way that the body gets rid of some drugs by directly eliminating them, or excreting them rather, by the kidneys. Thanks very much for watching. Look after yourself. Take care and see you later. Bye-bye.